Hi everyone, welcome to Baggy Infotech Media. I am KNS and today we are going to chat about the tools of genetic engineering, the part 3. We will be focusing on the nitty-gritty details like the tools used in the process and the cloning vectors. Let's dive in, come on. My next question to you, what are cloning vectors? Have you heard of cloning vectors? They are these tiny strands of DNA that don't cause any harm to whatever they are injected into. Scientists use them to identify and replicate certain parts of DNA. There are different kinds of cloning vectors like plasmids, podges, and others. They usually have a replication origin, a selectable marker gene like ampicillin or kanamycin resistance, and many cloning sites with different restriction enzyme sites. They are called as MCS, multiple cloning sites. One of the most famous cloning vector is PBR322, which Herbert Boyer made at the University of California, San Francisco back in 1977. Hey, did you know that circular double-stranded DNA molecules called plasmids, they exist separately from a cell's chromosomal DNA. They are naturally found in bacteria, yeast, and some higher eukaryotic cells and have a special relationship with their host cell. Plasmids can vary in size from just a few thousand base pairs to over 100 kilobases. And just like the host cell chromosomal DNA, plasmid DNA is duplicated before every cell division. Here are some examples of naturally occurring plasmids for you to check out from the following table. Plasmids are very interesting. You know, there are three different conformations that plasmid DNA can take: supercoiled, open circular, and linear. It is typically found in vivo as a tightly supercoiled circle that can only fit inside the cell. However, after plasmid preparation, some of the DNA may have single-strand nicks. If only one strand is broken, the DNA will still be circular but will allow rotation around the phosphodiester backbone, ultimately releasing the supercoils. Well, can you explain how plasmid DNA replication works? The blue strands are the original ones and the red ones are the new ones. The short segments represent the AT and GC base pairs that connect the complementary strands. The replication starts from the ORI and it proceeds around the circular molecule in both directions until the growing forks merge, forming two daughter molecules. The ORI is the only specific nucleotide sequence required to replicate the entire circular DNA molecule. In the end, we get two identical daughter plasmids. It's incredibly interesting that bacteria utilize plasmids to transfer genetic material within their own cells. There are five distinct types of plasmids such as resistance, virulence, degradative, core, and fertility F plasmids. Furthermore, plasmids hold significant importance in regulating gene expression within the cell. The next important thing you want to know is what are the requirements of a carrier molecule. If you want a molecule to work well as a carrier, it needs to have certain features. The kind of features you need will depend on what you are trying to do with the gene you have cloned. Some carriers have a prokaryotic origin of replication, which helps them survive in bacterial cells. Others have an eukaryotic origin of replication, which lets them replicate on their own in eukaryotic cells. To make it easier to build a library, most carriers have several unique cloning sites. And to figure out which cells have the carrier construct, they often include antibiotic resistance genes or other markers. Some carriers even have inducible or tissue-specific promoters, so you can control when and where the gene gets expressed. The most modern carriers have all kind of features that make it easy to clone, sequence DNA, mutate it in vitro, and replicate it on its own. Lactose operon, very important component in the cloning vectors. The plasmid is pretty neat because it helps with white and blue screening through alpha complementation. The only thing that seems a bit strange is that the DNA fragment is inserted into the middle of lag Z, which can make it inactive. Normally, lag Z codes for a protein called the alpha peptide, which is super important for the smooth functioning of beta galactosidase. But when this plasmid is used to transform lag Z deficient cells, it complements the sequence and gives it what it needs to work properly. Pretty cool, right? This slide further elaborates the lag Z gene. The beta galactosidase enzyme is coded by the gene Z, which can be divided into two parts, alpha and beta. Both parts need to be present for the enzyme to function properly. The combined activity of the two polypeptides is equal to the entire gene product. Certain bacteria with mutations lack the alpha part and exhibit no beta galactosidase activity. As a result, when grown with egg gall, their colonies remain white. However, when these bacteria are transformed by a plasmid containing the lag Z gene, which codes for the alpha part, the plasmid supplies the missing component and the colonies turn blue when exposed to egg gall in the culture medium. If a DNA fragment is introduced into the plasmid, it disturbs the lag Z gene and the colonies remain white. Few examples of the classic cloning vectors. Did you know that there are different types of classic cloning vectors like plasmids, pages, and cosmids? However, their size insert capacity varies and is limited. Plasmids can hold up to 10 kb, pages up to 20 kb, and cosmids up to 45 kb. Please check out the following table below that shows the corresponding insert size limit of each vector, and this is for your reference. Can we use bacteriophage lambda as a vector? Bacteriophage lambda was the first ever viral cloning vector introduced way back 1974. It's pretty cool because it can hold larger pieces of DNA than a plasmid vector, making it widely used for preparing genomic libraries, for cloning small DNA fragments, and the Punch lambda genome. Scientists use insertion vectors that have unique restriction endonuclease sites. These vectors are typically utilized for preparing cDNA expression libraries. When the recombinant viral particle infects bacterial host cells, It is called transduction. After Paj reproduction, the host cells lyse and release progeny virus particles. You can actually see these viral particles as clear spots of lysed bacteria or so-called plaque on an agar plate containing a lawn of bacteria. Each plaque represents the progeny of a single recombinant Paj and contains millions of recombinant Paj particles. Most contemporary vectors carry a lag Z gene, allowing for blue-white selection. Very interesting to learn about the life cycle and DNA replication of Paj M13. The M13 bacteriopage is a fascinating virus. It's made up of a type of single-stranded DNA that is about 6,407 nucleotides long. The virus is encapsulated in around 2,700 copies of the major code protein P8. Additionally, there are 5 copies of 2 different minor code proteins like P9, P6, P3, which are present at the ends of the virus. The life cycle is concerned. First, they undergo a conversion of single-stranded DNA to double-stranded DNA. In stage 2, they undergo rolling circle replication, and in stage 3, the core S protein, it switches the double-stranded DNA into the single-stranded DNA. Then the virus particles excluded from infected cell without lysis. Next important vector lined up is the yeast artificial chromosomes YARCs. YARCs can hold up to 500 kilobase pairs of genetic material and work as plasmids in bacteria when there is no foreign DNA present. When a fragment is added, they become like eukaryotic chromosomes and move to cells to replicate. YARCs consist of a yeast centromere, two yeast telomeres, bacterial origin of replication, and bacterial selectable markers. To create the YARC plasmid, yeast chromosome DNA is inserted into a unique restriction site. This cleaves the plasmid with another restriction endonuclease, removing a DNA fragment and causing the YARC to become linear. Once inside the cell, the recombinant YARC duplicates as a chromosome and foreign DNA is also duplicated. The next important vector to know is bacterial artificial chromosomes, BACs. Did you know that BACs can hold up to 300 kilobase pairs of data? It's pretty amazing, you know. And the F factor of E. coli is also impressive because it can manage large segments of DNA. Scientists use a process called electroporation to introduce recombinant bags into E. coli cells where they replicate like the F factor. One example of this is the P-BAC 108L. The regulatory genes ORES and REP-E help control replication of the F factor, while PAR-A and PAR-B limit the number of copies to 1 or 2. Plus, there is also a chloramphenicol resistance gene and a cloning segment included in box. The next vector we want to know is the cosmid vector. They are artificially constructed cloning vector containing the cos gene of Paj lambda. With the help of a special cloning method, researchers can create a vector that includes a gene from Paj lambda. This allows them to use Paj lambda particles to infect E. coli and clone larger DNA fragments using plasmid vectors. The DNA fragments up to 45 kb can be cloned into these cosmid vectors. These clones, called cosmids, work like plasmids and replicate similarly, but they may not be as stable due to their size and number. Have you heard of plasmids or plasmids? They are a type of cloning vector that combines the features of filamentous Pog M13 and plasmids. This hybrid vector can grow as a plasmid and also be packaged as single-stranded DNA in viral particles. Plasmids have an origin of replication for double-stranded replication and an F1 ORI that enables single-stranded replication and packaging into patch particles. Phosmid. They are a special vector created by the researchers that can help make libraries of cosmid-sized clones, which are about 40 kilobases each. We found out that when you put clones into phosmid vectors, they stay stable better than if you put them in high copy vectors. There is a fancy phosmid called PCC1-Phos that has both the E. coli F factor replicon and the ORI V high copy origin of replication. This means you get the stability of single copy phosmid cloning and the high DNA yields you get from cosmic clones. I have information on prokaryotic cloning and expression vectors, PET15B and PRZ vectors. These are the routinely used prokaryotic cloning and expression vectors. Just wanted to let you know that the PET15B vector has a his-tag sequence at the beginning, followed by a thrombin site and 3 cloning sites. Also, the T7 expression region on the vector map is in reverse because of the PBR 322 convention, as indicated by TB045. The PR set vector is created to enable high-level prokaryotic expression that is controlled by the powerful bacteriophage T7 promoter. The expression is triggered by the protein tag known as his tag 6X. An excellent choice for eukaryotic cloning and expression vectors is the PCM-V6 AC-GFP. It is a mammalian expression vector that allows you to combine an ORF with a C-terminal turbo GFP tag. This has numerous applications in studies based on fluorescence, plus it functions as an entry vector for the precision shuttle system. GFP means it's a green fluorescent protein which always expressed along with our protein of interest. That's what we can able to see our expressed proteins using hemidog, etc. Have you heard of linkers? They are little bits of DNA that can be synthesized chemically. These linkers are blunt-ended and have sites within them where they can attach to DNA fragments using DNA ligase. By using a restriction enzyme, the original DNA fragment can be cleaved, leaving behind sticky ends that are necessary for cloning. To make sure the fragments stick together, a large excess of linkers and a high concentration of the enzyme are used. After treatment with S1 nucleus, the cDNA produced contains single-stranded recessive termini that are filled in and made blunt using DNA polymerase I treatment before ligase treatment. In order to make cDNA sticky, we need to add restriction site linkers because of the blunt ends. We can use ligase T4DNA and perform blend and ligation to attach the restriction site linkers to both ends of the cDNA. Then we can use the same restriction enzyme that we used to cleave the vector to digest the linkers. After that, we can mix the cut vector DNA with the cDNA in the presence of DNA ligase. To clone, we just need to transform the mixture into an E. coli host, but we should keep it in mind that if the cDNA has the same restriction site as the linkers, it is possible for it to be cloned in pieces. Next one is adapters. They are short synthetic oligonucleotides and they have one blunt and sticky end, which can be ligated to the insert DNA to create a new molecule with the sticky ends. To avoid self-polymerization of the sticky ends, the 5' P terminal is dephosphorylated and used in 5'OH form. This prevents the ends from being ligated with each other by DNA ligase. However, a disadvantage of using linkers is that the DNA molecule may contain the restriction site. A homopolymer is a polymer in which all the subunits are the same. Example, a DNA strand made up of entirely deoxyguanosine, polydeoxyguanosine, poly-DG is a homopolymer. Another way to create a sticky end is by homopolymer tailing. The enzyme terminal deoxynucleotidal transferase adds a series of nucleotides onto a double-stranded DNA's 3'OH termini. Typically, 10 to 40 residues are added at each end. Frequently, poly-DC tails are added to the vector and poly-DG to the insert DNA. When the DNA molecules are mixed, base pairing occurs between the two. Tails probably won't be the same length. It can be repaired immediately, or the length may be stable enough to allow entry into bacteria where the gaps can be repaired. The advantages of these methods are it prevents reformation of plasmids. These are some of the references to get additional information. I also acknowledge Temple University, Philadelphia and Bharathidasan University, Tiruchirappalli. Thank you.